Bismillahirrahmanirrahim. Our second lecture, that is the pertaining to cerebellum. This is lecture 2. Yesterday we discussed certain features, the anatomical and physiological and the phylogenetic aspects of cerebellum. So today we are going to do the functional unit of cerebellar cortex. Now the learning objectives is by the end of this lecture you must be able to describe the functional unit of cerebellum, describe the synaptic inputs into the cerebellum, describe the Golgi cells, discuss the functions of cerebellum, and describe the clinical manifestations of. Now we'll be using this diagram in whole of our lecture to understand the functional unit of the cerebellar cortex. As you can see in this diagram, this is the functional unit of cerebellar cortex. You can see two type of let me take it. You can see two type of fibers over here. The first of the fiber is the climbing fiber. Here you can see this is a climbing fiber, and the second one is the mossy fibers. So from the climbing and the mossy fibers, all of the input is coming into the cerebellum through these type of fibers. The climbing fibers, they bring the input in the cerebellum from the inferior olive of the medulla. So the fibers that are coming from the inferior olive, they are called the climbing fiber. And the fibers that are coming from any other afferents, maybe brainstem and other cortical areas and other areas, these are called as the mossy fibers from any other area of the brain other than the inferior olive of the medulla. They are called as the mossy fibers. Now, you must know the differences between these, the mossy fibers and the climbing fibers, and what are their use in this functional unit of the cerebellum. So first of all, we'll do the climbing fiber. The climbing fibers, whatever you see in red means excitatory, and whatever you see in black here is the inhibitory. First, let's do the layers of the cortex of the cerebellum. From outermost to the innermost, we have the molecular layer, the Purkinje cell layer, because these have the Purkinje cells that have been drawn in black. These are inhibitory cells. Then you have the granule cell layer, because we have the granule cells, you can see them in red, means they are excitatory. And then you have the deep nuclei. You can see the nucleus over here, and from the deep nucleus you have the output that goes out of the cerebellum is only through these deep nuclei. Now we discussed certain nuclei, 4 or 5 types yesterday, the vestigial and all those, and we discussed their function. But here for this simplicity, we just would refer them to as the deep nuclei. So these are the layer of the cortex. Now coming to the climbing fibers, for example, from the inferior olive of the medulla, fibers they come. Now these climbing fibers, they go ascend up till the molecular layer. You can see the ascent of these climbing fibers through to the molecular layer, and while doing so they give excite the deep nuclear cell, the deep nuclei layer of the cerebral cortex. They excite them, and then the output signal is usually excitatory as seen in red. However, as they ascend up, they go up to the molecular layer, and in the molecular layer they also affect, or they synapse on the Purkinje cell. So this is the story of the climbing fiber. Now coming to the mossy fiber. The mossy fiber is basically afferent from any other part of the brain except the inferior olive. So the fibers, they grow and they go and they end up in the granule layer, the granular layer, on these granule cells, and they synapse with them. And please remember at this point that the climbing fiber usually releases aspartate at the synaptic endings, so it is excitatory, and the granule cells usually release glutamate, and this is also a excitatory neurotransmitter, so it would cause excitation. So the mossy fibers, they end up on the granular layer on these granule cells. Now the granule cells further, they make synaptic content, their dendrites go up into the molecular layer, and you can see they form these parallel fibers. They would divide into two T, like an inverted T, and some fibers would go in the right direction and some fibers would move in the left direction, so forming these parallel fibers and synapsing with the dendrites of the Purkinje cell. Now, these fibers, the action potential that usually occurs in the climbing fiber is a complex spike. For example, there is one climbing fiber for, let's say, 5 to 10 Purkinje cell, but once it is activated, it would activate the whole Purkinje cell layer for a prolonged period of time because there is a complex spike. Now, what is a complex spike? I will just try to draw it over here. A complex spike would be like you have an action potential, but it won't. This is a simple spike. You have an excitation and then you have an inhibition. It reaches threshold. There's action potential and then it goes back. This would be a simple spike that usually occurs in these mossy fibers. For a complex spike, for example, there is an action potential, but it won't come down like that. And as it is coming down, it would wave and wean up and down, and uh, the picture would be somewhat like this. So this is a complex spike. This usually occurs in the climbing fiber, and it releases aspartate. And the simple spike usually occur at the mossy fiber, and it releases glutamate. So now where were we? So the granule cells. Now within the granular cell layer, you also have. Let me take some other color in this. Let's go with green. We have a type of cells which are called as Golgi cells. So these are the Golgi cells. These Golgi cells, their dendrites go up into the molecular layer, and however, these granule cells, they activate these Golgi cells. But in what happens, what is the function of these Golgi cells? They usually inhibit these granule cells. These are activated by the granule cells, but they inhibit the cells a fraction of a second later. After their activation, the function of these Golgi cells is to inhibit the granule cells. So these are a type of cells which are also present in the granule cell layer. Then two other type of cells are also present, which are present in the, you can say, the molecular layer. These are the stellate cells. So I would draw them into a star. For example, this is a stellate cell, and then you have the basket cells. These are the cells. So these cells are also very important because they would inhibit these Purkinje cells. There is not only a single Purkinje cells. For example, let's draw here more Purkinje cell layer. For example, you have a Purkinje cell over here. I have drawn it in, try to draw it in black, and then a Purkinje cell layer over here. So what does these basket and the stellate cells do? They inhibit these cells because there are a lot of Purkinje cells in all of these layers. So this is what we read already about the lateral inhibition. It would inhibit this Purkinje cell, this Purkinje cells, so that the action potential that is traveling through this Purkinje cell is more focused. So this is a process which is called as a lateral inhibition, and the similar would the stellate cell would do. It would inhibit any Purkinje cells here and there which are not necessary but are lying there on their way. We don't want to lose our focus. Since the climbing fiber has already activated this Purkinje cell, so the deep nuclear cell and the Purkinje cell, so we want to go with that function. So the other cells would be inhibited. So these are the story of the climbing fibers. I told you it has a complex spike. It activates, it releases aspartate, it activates a deep nuclear cell, and then it all ascends up to the molecular layer with synapses on the dendrites of the Purkinje cell, and inhibits. And the neurotransmitter that is released here is GABA, and you know that is inhibitory. So then inhibitory signals are going to travel to the deep nuclear cell, and they are inhibited a fraction of a second later. So now coming to the granules, this was the action potential that was occurring in the climbing fiber. And this is a mechanism what we call as turn on and turn off mechanism. For example, a signal was coming in, excites the deep nuclear cell, and the output is a contraction of the muscle, but a fraction of the later, the climbing fiber, it activates the Purkinje cell, and the Purkinje cell then further inhibits the same muscle. Uh, as we said that, as we did the stretch reflex, we said that the agonists, they are excited, and the antagonist muscles are inhibited. So this is the pathway. This is also called as the turn on and the turn off pathway. that happens in control of fine movements or muscular contraction. Now, coming to the mossy fibers, I told you that mossy fibers, they usually end up in the granular cell layer. On the granule cells, the granule cells further ascend their dendrites and they form these parallel fibers. And in the granule cells, you have the Golgi cells, which inhibit the granule cells. And these granule cells, you can see the mossy fibers are also activating the deep nuclear cell. Both the climbing fiber and the mossy fibers, they activate, they cause excitation of the deep nuclear cell, and then the mossy fibers end up on granule cell, and the granule cell further goes up into the molecular layer. So we did this is the basic functional unit of cerebellum, and we did the turn on and turn off mechanism. Now, one thing to mention over here is that the Purkinje cell, they learn to correct the motor errors, and there is the role of the climbing fibers. For example, if you are performing certain activity for the first time, like playing a piano, a flute, or any other type of activity which you are doing for the first time, it is not that exactly on the spot. You don't achieve all the contractions of the muscle at the set time and at the set moment, because the climbing fiber is not trained for it. For example, it would cause only one action potential or two action potentials, or it would, for example, excite Purkinje cell in a limited amount. But as you perform that function, and your proprioceptors and your muscle spindles, they inform your cerebellum through the spinocerebellar tracts, which we discussed yesterday, that the movement that you did was not on the spot, or it has these errors. So what happens, these climbing fibers, they learn from it, and the next time you perform that movement, they are going to make certain changes, so as that the movement that you perform is on the spot, and it is as synchronized as intended. So this is the role of the climbing fiber. And how does it do it? One theory is synaptic plasticity, and the second one is that it causes more action potential. It would either inhibit or more of the Purkinje cell system, or the action potentials would be generated more excitation of these deep nuclear cells. So maybe by this process, especially the sensitivity of the Purkinje cells to respond to the granule cell excitation, the Purkinje cells and the granule cells, because you can see these granule cells, they go up, the fibers are going up, and they end also on the dendrites of the Purkinje cell, and they also inhibit the Purkinje cell directly, either the dendrites or the body. So by adjusting the relationship between the granule cell and the Purkinje cell, and the climbing fiber and the Purkinje cell, we learn to correct motor errors. So now much of the further presentation is mostly for you guys to go through it. We have discussed the functional unit already. Okay, so we have these three layers acquainted with it. So now this is again, here you can see all of the layers, the molecular layer, and then the Purkinje cell layer, and then the granule cell layer, and you can see the cells in it and how the connections they are making. You can see this is a stellate cell, the basket cells, which are widely dispersed in the molecular layer and important for lateral inhibition, then the Purkinje cell in the Purkinje cell layer, and the granule cell in the granular layer, and of course the mossy fibers, which comes from any climbing fibers. So now this is a short diagram that shows different cerebral inputs and output. The output is usually excitatory, followed by a fraction of a second later by inhibition from the Purkinje. So now what are the differences? This is a very important exam question, the differences between the mossy fibers and the climbing fibers. So mossy fibers are all olivocerebellar, all except the olivocerebellar tract, and the climbing fibers are only the olivocerebellar tract. The mossy fibers are weakly excitatory because they have a simple action potential. They are strongly excitatory because they have a complex spike. The action potential is sodium dependent and the climbing fibers is calcium dependent. Effects short lasting, they are long lasting. Action potential, of course, is of small amplitude, it is large amplitude and it follows for a larger period of time. Action potential is graded, it follows all or none. Either there would be an action potential excitation, for example, the rapid contraction of the muscle, or there would be none contraction. So neurotransmitter usually not known, but glutamate we usually assume is the neurotransmitter for mossy fibers and aspartate for the climbing fibers. So there are 80,000 to 2 lakhs type Mossy fibers supply 1 Purkinje cells, and the ratio for the climbing fibers and the Purkinje cells is usually 1:1, though it can be 1 climbing fiber for 8 to 10 Purkinje. So all of that we have done already is the cerebellum, and we said that Purkinje, they are the prominent nerve cell, and they are named after this Czech anatomist Jan Evangelista Perkine, and it is found throughout the cerebellum, and they release GABA. That therefore now, there are medical conditions in which we can damage the Purkinje cells. For example, toxic exposure to alcohol or lithium, autoimmune diseases, genetic mutation, autism, or neurodegenerative disease, degenerative changes. And by doing so, our different actions, different contractions would be distorted, because the turn on and turn off cycle is. So now the coordination between the motor cortex and the cerebellum. When we were doing the spinocerebellar tracts yesterday, we discussed that from your proprioceptors, the muscle spindle, the joint proprioceptors, the Pacini corpuscles, and from all those your sensory organs, the signals are continuously transmitted into the cerebellum, and cerebellum is apprised of what movements you want to do, and there is an image is formed of the intended movement. And then through the both the dorsal and the spinocerebellar tract, how the signals they pass into the cerebellum, and then for the different appraisal of the cerebellum of the periphery and your proprioception, it is interlinked. You can see whenever from your motor cortex, directly from your cortex to the muscle, we have this corticospinal tract. This is also very important, and important different types of lesion that occurs in the corticospinal tract. For your exam, it is also very important, so you must go through this tract. I did send a PowerPoint, but that was not recorded. I would just try to, if we could meet on Google Meet or Zoom, so that we can do this corticospinal tract. It is very important. So the fibers, when they are moving down directly from the cortex, you can see the connection. They go into the intermediate zones of the cerebellum, and it is apprised that this movement is going to, that this action is going to be taken place. So cerebellum is all the time being working on the background of the what action is going to take place, and where your body is, where your different parts of the body, the muscles, the joint, your limbs are at a different point of time. So now the general function of the cerebellum, we split the functions of the cerebellum according to different lobes, anatomically, physiologically, and phylogenetically yesterday. So these are just the gross the what function, that is the planning, coordination, and posture maintenance. And then mostly due to damage of the deep nuclei, for example, the dentate nucleus, there are different manifestation of cerebellar regions. There are different tumors, hemorrhage, cerebellar infarction, or motor disease, for example, disorders, lesion in the vermis, in the flocculonodular lobe, in any part of the cerebellum would lead to certain clinical manifestation of the different diseases. So the clinical features usually occur on the same. Now we'll do the cerebellar region, for example, coming to the posterior or the flocculonodular lobe. Of course, there would be postural and gait dysfunction. Then in the paliocerebellum or the vermis, there is truncal and gait ataxia, if you remember the homunculus of the vermis. And the hemispheres, the lateral hemisphere, that is the neocerebellum, usually with the limb atraxia, the dysmetria, dysrhidokinesia, intention tremor, dysarthria, and hypotonia. Each one of them, I'm going to define it in the later slides. So this is, you can see in the first one, the A, you can see this is a normal type of movement, for example, and this is the delay. For example, from here you have a threshold potential, and then you go upward. But in an abnormal movement, what would happen there, that there in initiating the movement, for example, the turn on signal is so weak, if there is a problem in your cerebellum, or there is cerebellar lesion, so there would be a delay of a fraction of the time before you go, and if the movement then does occur, you can see what kind of movement it would be, because the action potentials, they are not as regular, they are going up and down. So for example, if you ask someone to touch a spot and then touch your nose and then go back to that spot and touch your nose, or finger nose test, this would be a normal person. But in an abnormal, you can see, you can just touch this spot and then you can go back to your nose. But what would happen in someone with cerebellar disease, he would touch that spot, but we would be wandering here and there because he doesn't know of the proprioception is not working, and coming back to his nose, he would just linger on like for ages. So this is what happens usually during a normal, if you, for example, clapping your hands or doing movements which are fast enough, this is what happens. This is what type of spike usually form. And in abnormal, for example, in dysdiadochokinesia, in which this is the inability of a person to perform rapid alternating movement, this is what kind you can expect. So all these 11 are the different clinical abnormalities that you see in a patient with cerebellar lesion. First to hypotonia, this is the most characteristic lesion, and usually hypotonia is on the side of the lesion. And a person with cerebellar disease shows no abnormality while at rest, but abnormalities become as soon as performing certain type of a movement. Now, attitude, you can see that by looking at the patient, you won't be able, he would not look much normal, because face is rotated to the normal side, shoulder on the affected side is slightly raised, and in the opposite side, cutting it in short, a person is not able to maintain his posture probably, and because of that, he has dysmetria, inability to place an extremity at a precise point in space because of the loss of all those tracts which apprise you about your extremities, about your limbs, about the contractions of them, and where they are in the present, whether you want to flex them, rotate them, you just lose that ability. And there is loss of sense of distance. For example, in monkeys who had these cerebellar disease, if a normal monkey was asked to go and run from point A to point B, and the point B is a wall, so a normal monkey realizes that I'm nearing the wall, and would step a few seconds before, would stop running because of the danger of just going into the wall. But the monkeys that had cerebellar lesions, when they start running, they won't realize their relationship to the nearing wall, and they would just go and bash their brains on the wall, and they would not stop. Like they are not apprised of when to stop, when to start. That is just so this is a simple, the finger nose test I told you already. For example, you touch a spot A, and then you go back, ask the patient to touch his nose. So a normal person can do that easily, but a person with cerebellar disease, you can see here, you'll be going here, there, and then finally going to the nose. And sometimes it's not even nose, may poke his finger into his eye or some other. Dysarthria is defective speech. Of course, for speaking, for articulation, there is a relationship maintained between the agonist-antagonist muscle, and there is vocalization. This is also distorted, and such a person is usually having a scanning type of a speech. Or you can say slurring, even decomposition of the movement. The patient decompose movements into component parts, cannot measure rate. I told you direction, I told you, and extent of the force point. All these three would be distorted: the rate, the direction, and the movement. And then you have the intention tremors on movement, like drinking a glass of water. Now this is important to cerebellar disease, that is intention tremor. In different CNS type of disorders, sometimes you have tension, these tremors at rest, even in certain diseases, as we have done. But particularly in cerebellar disease, you have as soon as you, for example, want to pick up the glass of water, you will start. Then you have the dysdiadochokinesia, inability to make rapidly alternating or successive movements. And then you have the rebound phenomena elicited by asking the patient to flex his forearm against resistance. The resistance is suddenly removed. If it is removed, for example, you were resisting, you had your hand on it, and when it is removed, the limb overshoots the normal part. So there's overshooting of the different muscular movements because cerebellum is usually damps the different types of muscular contractions. Where needed, it would cause excitation, and it would there, for example, you saw there is lateral inhibition, and then there is inhibitory cells Purkinje, which inhibits. So there's usually damping of different muscular movements, and when cerebellum is not working properly, this damping does not occur. So you usually overshoot your normal range. And then tremor of the eyeball, your eyeball, even when you are moving it, since it is also a movement, would not be able to, for example, you are driving in a car and then your eye has to fix on different moving objects because you are moving. And then with your movement, your eyes fixes on different, for example, trees, and then boards, or a person driving by, or a person in a nearby car. So you do that very swiftly. But if somebody with cerebellar disease, then it is very difficult for that person and to just focus, and there are tremor in the eyeball with movement. And cerebellar ataxia, the last one, in coordination of the muscular action. For example, during walking, the synergy between the different agonist muscles, and then then their application, and the antagonist muscles. It is so. This is diagram showing like you are just having a vertigo type of thing here and there, and you just cannot walk. Then there are, of course, there is this disturbance of the posture and the gait, and usually the head is usually tilted to the side of the lesion, and gait is unsteady. For example, in a drunken fashion. Now, whenever you have nystagmus and you have intention tremor and you have dysarthria, we call this as Charcot's triad, and this is diagnostic for somebody having disease, this cerebellar disease. And intention tremor is the hallmark of cerebellar disease. You must. Now again, a second assignment. I didn't get any assignment that I posted yesterday. So this is your second assignment. These are SEQ type of question. I want you guys to do some workup, and then make these in Word document. These are like short essay question, or you can even write them and take the photos and then upload them. So these are the question. Describe the effects of cerebellum on the movements to make them smooth and to the point. And then you have to define the following terms. What is dysmetria, pass pointing, dysdiadochokinesia, and nystagmus? These are exam questions. These are the questions that are usually asked in OSPs, and then you don't know them, so you just have to define them. Define them in a single line at least. Name the function of cerebellum and discuss pass pointing. Now you have to discuss why and how pass pointing occurs and what circuit is implicated in it. Name any 4 cells of the cerebellar cortex and write their functions. So we did a lot of cells today. We did Golgi cells, we did granule cells, we did deep nuclear cells, then we did the Purkinje cell, then we did the basket cell, the stellate cells. So from these, any 4 important ones, you can just name them and then write their functions. So this is the end of our lecture, and here we just complete our cerebellum part of the motor physiology. Thank you. See you soon.